So today I'm going to tell you about work that we've been thinking about in my lab in the last few years, in which we're trying to develop conceptual models for the ways in which genetic variation maps to complex traits. And this is joint work with Evan Boyle, who is a graduate student in my lab, Yang Li, who is a postdoc in my lab until recently, and he's now an assistant professor at the University of Chicago, and Chuanyao Liu, who's also a colleague at the University of Chicago. So, during the last dozen years or so, we've had a revolution in our ability to study the links between human genetic variation and complex traits through the advent of genome-wide association studies. And in many ways, genome-wide association studies have been a real triumph, as we've now been able to identify many thousands of variants that affect a wide variety of different human quantitative traits, phenotypes, and diseases, and these are replicatable signals. And what we understand now about the genetics of complex traits has really been transformed from where the field stood perhaps a dozen years ago. But today I'm going to talk about some things that we have. Some of the general patterns that have emerged from this body of work that I think require further explanation. And so, first of all, I'm going to argue that the heritability of typical complex traits is driven by really huge numbers of variants with very, very small effect sizes. Secondly, that most of the heritability is mediated through genes that are not directly related to the trait of interest. And I think that we really need to have new conceptual ways of thinking about the molecular processes that link genetic variation to complex phenotypes. And if we sort of go back a dozen years to the beginning of the complex trait mapping era, I think that people thought that complex traits would be somewhat like monogenic traits, but a little bit more complicated with smaller effect sizes. And I want to argue that in some ways they're really fundamentally different, and we need to have frameworks for thinking about this. And I'll suggest one framework that we've been developing in my lab. So first of all, let me talk you through a couple of examples of traits that I think are illustrative of this. So the first of these is for schizophrenia. So in the latest meta-analysis for schizophrenia, which was published in 2014 and had about 150,000 individuals, there were 108 genome-wide significant loci. So these are all these sets of loci that are above this red line here. So that's a remarkable achievement to be able to identify so many loci that are contributing to this disease, which has been, prior to that, incredibly hard to study. But one thing that's been really extremely interesting about schizophrenia and many other complex traits is that these significant loci only contribute about 10% of the expected heritability. And 8 or 10 years ago or so, this was referred to as the mystery of missing heritability, and the mystery has largely been explained by understanding that there's actually an enormous number of additional signals with very, very small effect sizes that sit down here in the weeds that are not significant in current sample sizes. And this has been through work of many people. Peter Vischer in particular sort of pioneered work in this area, and in the case of schizophrenia, this has suggested that all of the genetic variation together, all of the common variation, explains about 80% of the heritability. It's just that most of that is driven by an enormous number of variants with tiny effect sizes. Okay, so you might say schizophrenia is a very complicated disease. We don't really understand it. So what about a potentially simpler trait that we know a bit more about the biochemical basis of? Next, let's talk about lipid levels for a moment. So these are traits like LDL and HDL cholesterol and triglycerides. So we actually know a lot about lipid metabolism and the processes that determine lipid levels from monogenic studies of dyslipidemias and familial hypercholesterolemia. So there's a review from a couple of years ago that lists about 24 major effect loci that affect lipid levels. And here's a set that they list for high and low LDL cholesterol levels. So they list about 10 genes here for LDL levels with monogenic, very, very large magnitude effects. on LDL levels. Okay, so that's describing the effects of monogenic disorders in LDL levels. So what about common quantitative variation in lipid levels within individuals? So you can do GWAS for that, and so here in GWAS the target is really uncommon variation. And there have been quite a lot of studies in this area, but this is a plot from a very nice study by Kristen Willer and colleagues in 2013, where she reported 57 genome-wide significant loci that affect different aspects of lipid levels. So what's the overlap, you might ask, between the common variation affecting lipid levels and the monogenic traits? So in the case of LDL cholesterol, we can go back to these 10 genes that have been associated with monogenic disorders. And in fact, 7 of those 10 also pop up in the genome-wide association studies. So it's not the same variants, right, because these variants in the monogenic traits are extremely rare mutations, but most of these genes are also popping up, usually through non-coding variation that's in the vicinity of those genes, presumably affecting regulation of those genes. Okay, so most of these big hits are coming up in the GWAS. There's sort of core genes for affecting lipid levels. So should we expect that these are explaining a lot of the variance in lipid levels? Well, actually, the significant loci together, so 36 significant loci together for LDL cholesterol, only explaining about 20% of the heritability. But if you put all of the genetic variation together, that explains about 80%. So again, even in a trait that in some ways is biochemically much simpler than something like schizophrenia, most of the variation is coming from a very, very large number of loci spread widely across the genome. So today what I'm going to do is talk you through first a set of 3 observations that I think are particularly pertinent when we want to think about the architecture of complex traits, and then I'm going to describe to you the model that we've developed for trying to put together everything into one framework. So first of all, as I've already hinted to you, for many traits, heritability is spread extremely widely across the genome. So a number of groups have done work on trying to estimate how much of the heritability comes from individual chromosomes. So this is a plot from Bogdan Pasaniuk's lab, in which they're estimating how much heritability comes from each chromosome for schizophrenia. So each of these blue bars here is an estimate for each of the chromosomes. And so the y-axis is heritability, the x-axis is thousands of SNPs, so it's essentially a measure of how big the chromosomes are. And you can see that you can predict how much heritability is coming from each chromosome simply from chromosome size. So this should immediately suggest to you that there must be an enormous number of variants across the genome that are contributing without large loci without loci of big effects that would sort of create outliers. There is a small outlier here, which is chromosome 6, and this of course contains the MHC region. There are two of the largest hits in the genome are in the MHC region. How does this look for a complex trait that does have a big effect locus? Well, this is what rheumatoid arthritis looks like. So there's a huge effect in the MHC region, but you can see that that's actually on a polygenic background. And this is actually a very general kind of pattern. So even in developmental disorders that we usually think of as being driven by very large effects, often de novo mutations, there's new work from Matt Hulse's lab that shows that this is actually sitting on a polygenic background that's predisposing and spread really genome-wide, involving many loci. Okay, I just got a message. Another device on your network is using your computer's IP. Okay, I'm all right with that. Okay. Okay, so that's what it looks like at chromosome scale. What if we try to drill down to finer scale to understand how much the genome is contributing? So this is a little bit difficult for a couple of reasons. First of all, we need to try to measure the contribution of loci that are not actually significant, genome-wide significant. Secondly, there's this issue that genome variation is structured into blocks of linkage disequilibrium. So in the cartoon model, you can kind of think of these as haplotype blocks. So if you have a causal site, then not only it will show a significant association, but all the variants that are in LD with that will also tend to be associated. Okay, so to tackle this problem, one thing that we can do is to look at the amount of signal on SNPs as a function of how many LD partners each SNP has. So the approach that we've taken to this is, first of all, to divide is to determine for each SNP how many LD partners it has. This is sometimes known as LD score, so you can think of this as being something like the haplotype block size. And then secondly, for SNPs in each category, we want to estimate what fraction of those SNPs have an association test that's drawn from the null hypothesis and what fraction from the alternative. So this is analogous to what's done in false discovery rate analysis. So if you're familiar with John Story's pi1, that's a method for trying to estimate what fraction of tests are from the null and from the alternative. We actually use a method that Matthew Stevens has developed called ASH-R, which has actually got some properties that make it better in some ways. Okay, so if we take the lowest 1 percentile bin of SNPs in terms of LD score, so these are SNPs with the fewest LD partners, then we estimate that about 10% of these have a non-zero association with schizophrenia. Okay, now let's take that out across the whole distribution, and this is what this looks like. So each bin here each dot is a bin of 1% of all of the SNPs, and what you can see is that we're estimating that for the largest bins, more than around 2/3 of these actually have a non-zero association with schizophrenia. And overall, we estimate that about 57% of SNPs are linked to a variant with an effect on schizophrenia. So this should immediately imply to you that there must be an enormous number of SNPs across the genome that are affecting schizophrenia. And we estimate that there's something like 2% of SNPs in this analysis have causal effects. And, you know, there's some imprecision in these estimates, but you should take from this that there are an enormous number of variants spread very widely and sort of thinly across the genome, very almost uniformly across the genome that affect this trait. And we get similar results for height. So for both of these traits, we've got something like 50% of haplotype blocks affecting the trait. And if we take the typical size of haplotype blocks, this suggests on the order of 100,000 causal variants. And this should suggest to you that, you know, most kinds of genes are implicated in affecting these traits. Okay, so here's a second kind of observation. So many different groups have shown that GWAS signals tend to be highly enriched in chromatin that's active in cell types that you know, kind of make sense in terms of what the trait is. I'll give you some examples of this in a moment. But, you know, for example, you might think of CNS tissues for schizophrenia. And people have interpreted this in the literature as meaning that these are signals that are driven by cell type specific regulation. But surprisingly, perhaps, it doesn't actually matter whether the chromatin is broadly active. So think, for example, of promoters for housekeeping genes that are active in every single cell type, or, you know, what we would think of really as being, you know, sort of very narrowly specific open chromatin that's being turned on to regulate, for example, T cell function. Okay, so so first of all, I want to convince you that heritability in general is highly enriched in active chromatin cell types that make sense. So this is a plot from a paper by Hilary Finucane in Alcus Price's lab. So each plot here shows the enrichment of heritability in different in active chromatin maps for different kinds of tissues for a set of different traits. So, for example, for bipolar disorder, the strongest enrichment is in CNS tissues. For height, the strongest enrichment is in connective and skeletal open chromatin. For fasting glucose, it's pancreas and so on. So these kind of make sense. These are sort of what we should expect for these traits. And as I suggested, you know, from first principles, I certainly had expected that what this meant was that these were signals that were driven by cell type specific open chromatin. So let's try to test this. So we could classify the chromatin that's open in the most important cell type into different categories. So we could think of chromatin that's uniquely active in the most important cell type. So for example, if we're looking at an autoimmune disease, we might look at chromatin that's only active in immune cells versus chromatin that's somewhat specific, so it's open in immune cells, but perhaps also a couple of other tissues, or chromatin that's broadly active, so meaning that it's active in most tissues of the body. So we can ask whether there's a difference between those categories in terms of how much they contribute to disease risk on a per-SNP basis. And perhaps surprisingly, there's not much. So this is an analysis of the relative heritability that we see coming from SNPs in each category. And this is for 3 different traits here, for schizophrenia, which is associated with neuronal tissues, rheumatoid arthritis and Crohn's disease, which are associated with immune cell types. Okay, so now if you compare the heritability on a per-SNP basis for uniquely active chromatin versus broadly active chromatin, you can see that these are roughly similar. They're not exactly identical, but it's certainly not the case that all of the signals coming from these uniquely active open chromatin regions. And of course, then there's an important negative control, which is chromatin that's not active in the most relevant cell type is contributing very, very little signal. So this is really arguing against the model that the data are completely noise and just overall spurious. Okay, so that's the second observation. The third observation is that while a number of groups have shown that if you look at rare variants or the very strongest hits, so rare variants often have large effect sizes, these do often identify core genes and core pathways that kind of make sense. But most of the heritability is really very broadly distributed across the genome and broadly distributed across different geocategories. And the main requirement is that the genes do have to be expressed in the right cell types or right tissues. So let's look at this for schizophrenia. So these are so what we're doing is classifying genes by gene ontology categories, and so each dot here represents a different gene ontology category. That the x-axis is the proportional of all SNPs that fall into that geocategory, and on a log scale, and the y-axis is the proportion of heritability that's estimated to come from SNPs in or close to those genes. And what you can see is that you can actually predict pretty well how much each geocategory contributes to heritability based simply on the number and size of genes in that category. There is a little bit of signal here, so basically distance off the diagonal is a measure of signal. Calcium ion channel activity and ion transport genes, which are involved in neuronal activity processes, those categories are sort of among the furthest off the diagonal. So that's sort of comforting. We see kind of an enrichment for transcription factors, but overall the category that's contributing the most here is a relatively generic signal from protein binding genes. So the overall pattern that you get here is one of simply the size of the category is what's just determining how much it contributes. And then if we look at Crohn's disease, which is an autoimmune trait, then we see that the most enriched categories flip a little bit. So immune response genes are among the most enriched, while neuronal genes are depleted. But these are relatively weak signals. Okay, so to recap, for a wide variety of traits and diseases, we have first of all that heritability is spread very widely across the genome. We have that genes with trait-relevant functions only contribute a small fraction of the total disease risk, although if you look at variants with the largest effect sizes, these do often have clearer enrichment in the relevant gene sets, indicating probably that they're sort of biologically more proximate to the mechanism of disease. And then thirdly, we have that contributing variants are really highly concentrated in regions that are active chromatin in relevant tissues, which probably implies that most effects are coming through gene regulation, but they're not particularly enriched in super specific open chromatin that's sort of involved in the specific functions of cells that are most relevant to disease. So I want to stop for a moment now, and I want you to think, how do we conceptualize the molecular mapping from genetic variation in the genome to complex traits, given the things that I've told you? And I'm going to suggest one model for this, and we can talk about this. So we have a paper, a perspective piece that we published last year, in which we proposed something called the Omnigenic model, and so the Omnigenic model suggests that there are three classes of genes, and the first type of gene are what I'm going to refer to as core genes, and these are genes that have direct effects on the phenotype. So if you think about the example of lipids, the monogenic genes are generally genes that have sort of a direct role in the biochemistry of lipid metabolism, or import or export of LDL particles in and out of the bloodstream, things like this. Okay, the second set of genes are what we refer to as peripheral genes, and so this will encompass the set of all other genes that are expressed in relevant cell types. So a very, very large category. And what we propose is that there's an enormous number of trans-regulatory effects that come from these peripheral genes. And then the third category of genes that are not expressed in the most relevant cell types, and these actually really don't contribute to disease risk. And one really important point of this is that what we think is that most of the phenotypic variance is coming from regulatory variation that's initiated through peripheral genes. So core genes are only explaining a very, very small fraction of the heritability. And so one way that I like to think about this is that, just to make some numbers up, let's suppose that for some trait you've got perhaps 100 core genes that have direct effects on the trait, and there's on the order of 10 to the 4 genes that are expressed in the relevant cell type. So this means now that the peripheral genes are outnumbering core genes by something on the order of 100 to 1. And so what we think is that the peripheral genes dominate the phenotypic variance through sort of weak regulatory effects that ripple through shared gene regulatory networks in cells. Okay, so that's sort of the model that was outlined in the perspective piece last year. And in the second half of the talk, I want to tell you a little bit more about our newer thinking on this, and trying to sort of be a bit more specific about precise models in the context of what we know from, in particular, cis and trans EQTL studies. Okay, so now, first of all, imagine that we've got a core gene, and my definition of a core gene is that it has there's a direct causal arrow that we can draw from the gene product to an organism level phenotype. Okay, so we could have so how does the core gene affect the phenotype? Well, first of all, we could have missense variants inside the core gene. That certainly happens, although we know that those contribute only a small fraction of the heritability. Secondly, we can have cis-acting regulatory variation, and we think this is an important part of the story. And then third of all, we can have trans-acting effects that are coming from all of the other expressed genes in that cell type. So these are trans-acting effects from peripheral genes. So how much of the expression variance is due to cis versus trans effects? So in general, we don't know yet what are the core genes, but we can at least answer this question for genes in general. Okay, so one thing that I've done is to go back through the literature and try to find as many papers as I can that have clear estimates of cis versus trans contributions to heritability. And this has been done in a wide variety of different organisms and using a whole range of different statistical methods. In general, this is a somewhat hard thing to estimate because trans effects are small. But what we find from the literature is that the estimates, although there's some variation among them, that they fairly consistently range from estimates of about 60% of the heritability of gene expression coming from trans effects up to about 90% of the heritability. So the majority of expression variation that's genetically determined is coming from trans effects. So just to put a number on this for the sake of convenience, I'll say for the rest of the talk that something like 70% of the heritability of expressions coming from trans effects. Okay, so now we can put some numbers on this plot. So about 70% of the heritability for a core gene would be coming from trans effects from peripheral genes, and 30% from from heritability from cis, if we make the assumption that the core genes are perhaps typical of, or perhaps typical of genes in general. Okay, now if anybody who's tried to look for trans-EQTLs will know that trans-EQTLs are very, very hard to find. And this is because they have very small effect sizes compared to cis. And so part of the challenge is because of multiple testing, but that's actually not the main thing. The main thing is that trans-EQTL effect sizes are tiny. So this is a plot from our collaborator Chuanyao Liu, and what she has done is she's taken all of the genes in the genome, and for each gene she's asked what's the biggest effect size in cis and what's the biggest effect size in trans, and then she's looked at the, and then you have some winners cursors. She's looked at the size of those replicated in a second data set. And so this is a cumulative plot of the effect sizes of those SNPs on a z-score scale, and so you can see quickly that the effect size of cis-EQTLs are just huge compared to the effect size of trans-EQTLs. And in fact, when we're interested in variance, what we should do is actually square the y-axis here. And if we did that, you wouldn't even be able to see the trans-EQTLs coming off the line at all, which is why I just show it on the z and not z-squared scale. So, okay, so we've got 70% of the variance in expression coming from trans effects. We've got trans associations being much weaker than cis effects in general. And so together these imply that even though we can't find them yet, it means that a typical gene must have huge numbers of very weak trans-regulators. And so if we imagine now that there are perhaps tens or hundreds of core genes for a typical trait, now this model starts to explain how it can be that a large fraction of the genome is contributing to any given complex trait. Okay, so that's maybe answered one of the key questions. So under this model, we now have some idea of why so much of the genome contributes. But a second question is, why do the core genes not contribute more? So why do we not see more big effect sizes at genes that we think should have important roles? So one possibility would be that core genes tend to be under stronger purifying selection, and I think that may be true in some cases. But this is really a universal observation among almost any kind of trait that we look at. So I'd really like to think of more general sorts of explanations. And so I'm going to suggest one model that can explain this. And now to do this, we need to start with writing down a simple phenotype model. And so I'm going to suggest basically the simplest model for phenotypic variation that we can have is based on a linear model with the expression that's looking at the expression levels of the core genes. Okay, so now, first of all, let's imagine we've got some quantitative phenotype y sub I. So this could be either something like LDL levels, or if you're more interested in a disease like diabetes or schizophrenia, this could be some underlying liability score. Okay, so we're going to model the phenotype in an individual as being equal to a sum of the average phenotype plus a sum over m core genes of terms that have got the following in them. So we're going to look at the expression level of core gene J and individual I, and we're going to look at the difference between that and the mean expression of that gene in the population. Okay, so this is the deviation, the individual level deviation and expression for a particular individual and gene. And then we're going to multiply that by times a scale factor that measures how much effect that gene has on the phenotype when you change its expression level. And then lastly, we have a random error term that's going to include environmental effects, stochastic effects, and so on. Okay, so now what we want to do is to compute the variance of the phenotype. So this is so this is the additive variance, and so variance of y sub I. This is going to have two parts to it. So first of all, we've got a sum of variance terms. So this is the variance in expression of each gene, of each core gene J, times this the slope squared. So there's m of those terms, plus a series of covariances. And so each of these covariances is going to be a covariance in the expression of two core genes across individuals times the product of the slopes. And then we have a variance term again. Okay, so now we can start to think about how these are going to be determined by cis and trans effects on the core genes. Okay, so the variance in expression of each core gene, I suggested to you before that that should be something like 1/3 cis effects and 2/3 trans effects. And the expression covariance, what we think is that these are actually dominated by trans effects, i.e. peripheral genes. And so the intuition for that is simply that individual trans effects are small, and so it's unlikely that you're getting a lot of covariance that's coming from the partner. Most of it has to be coming from other genes in the network. Okay, now the key thing here is that there are m of these variance terms, and there are almost m squared of these covariance terms because they're comparisons between all pairs. Okay, so now I'm going to suggest two extreme versions of this core gene model to illustrate that. So first of all, let's imagine a setting where the expression covariances of the core genes are low. So you can think of this really as being a situation where the core genes are really far apart in the regulatory network. Now, in this case, we've got core genes, and they have a direct arrow to the phenotype, and each core gene is going to have its expression affected independently by all of the other peripheral genes in the cell. And so in the context of this, of the math, what I'm going to assume here is these covariance terms are 0. And so now basically when we compute how much of the heritability is coming from effects that are close to core genes, then we would expect that about 30% of the so 30% of the expression variance is coming from cis, 70% is coming from trans, and overall we would predict that about 30% of the heritability is coming from the neighborhood of core genes. So I would argue that this actually really doesn't look like the data at all. Where I showed you for even for lipid levels, where we've got a pretty good idea what the core genes are in the most, in a quite expansive set of genes, that's only explaining about 20% of the variance. Okay, so now let me suggest a second model where the core genes are tending to be close together in networks. And what this will do is it'll mean that the expression covariances tend to be high. Okay, so here we have these two core genes are sitting in the same co-regulated network, and so I'm going to imagine here that the effects from peripheral variation tend to be shared rather than independent. And so now these covariance terms are going to tend to be usually positive, but anyway they'll be non-zero. And there's one more condition, which is I'm going to assume that these slope terms tend to be pointing in the same direction. And I think this actually kind of makes sense. So if you imagine perhaps inflammatory processes in immune cells that lead to autoimmune disease, then you might imagine that you've got these regulatory cascades that kind of promoting inflammation, and and there's a whole set of genes that are all involved in increasing inflammation and immune response, and each of those genes tend to be co-regulated, and they'll all on average tend to increase risk, if anything, they'll tend to increase risk of autoimmune disease. So it's not unreasonable to think that these gammas should tend to align in their signs. Okay, so now the cis effects tend to be independent for the core genes, but the trans effects are often shared across core genes, and what this means is actually most of the heritability winds up being transferred out to peripheral genes, basically because these m-squared covariance terms are dominating the heritability. And another intuitive way to think of this is that basically the cis effects are kind of, on the core genes, are kind of idiosyncratic for any individual. So some are pointing up and some are pointing down, increasing or decreasing a risk, but the peripheral effects are all shared, and so they all kind of sum together and add into this squared term. And so that's why we think that these peripheral effects are dominating under this model. Okay, so for the last few minutes, I just want to talk a little bit about the other side of the problem. So far I've been taking kind of a core gene-centric view of the world. So how does variation funnel through core genes? So the other important question is, what are the effect sizes of individual SNPs? Because when we're doing mapping in a GWAS study we're measuring SNPs and not core genes. So, and then secondly, if we look at the genes, if we look at the SNPs that are at the top of the list, how interesting do we think those are likely to be? Okay, so let's just start with the simplest setting. So let's suppose we've got a core gene, it's got a direct effect on the phenotype, and it's got a cis EQTL. So I'm going to say that beta sub ij is going to be the effect of SNP I on the expression of gene J. And I'm also going to assume that a change in expression of gene J has an effect gamma J on the expected phenotype. So now the effect size of this SNP on the trait is going to be beta gamma. Okay? And then if we're interested in variance, then it's beta squared times the allele frequency times 1 minus the allele frequency. So basically the variance will be proportional to beta gamma squared. Okay, so we know that cis effects can in many cases be quite large, so this beta gamma term can be a relatively large effect, at least in the scale of complex traits. Okay, what about an individual trans effect? So now we can again have an effect beta ij, which is the trans effect size of this SNP I on core gene expression. But we know that the trans effects individually tend to be tiny. And so when we compute beta gamma, these are almost uniformly going to be very, very small effects that are going to be difficult to see. But trans effects, now these are potentially affecting many genes. And this is particularly going to be the case if we're looking, for example, at transcription factors that are master regulators. So now imagine that we've got a SNP here that is a trans-QTL for a whole set of different core genes. So from this SNP I, we're going to have a beta ij to each of, where j is subscripting each of these different core genes. So there's a different beta ij for each core. And then we're going to have a different gamma j from each core gene to the phenotype. So now, when we compute the effect size of this SNP on the trait, we now have a sum of these beta gammas. And so if the beta gammas tend to have the same sign, then and there's enough core genes that are being regulated, so now this could actually add up to be important. So now the effect size is going to be the number of core genes m times the average of the beta gamma. So the trans effects individually are small, but they can add up to be a lot. And now the last setting would be a transacting factor that does affect expression of quite a lot of core genes, but the effect directions are random. Okay, so sometimes it's increasing expression, sometimes it's decreasing of expression. So the signs of the beta gammas are sometimes positive and sometimes negative. And so in that case, the expected value is 0, but you've got sampling. And so actually these can wind up being non-trivial. So the standard deviation of this is equal to the square root of the number of genes that are regulated times approximately the average of the beta times gamma. So sometimes a variant that's a trans-EQTL with effects on many core genes will actually have non-trivial effects. And I think that when you look across the whole genome and find that there's an enormous number of variants that have non-zero effect, it's because typical regulatory SNP may have non-zero effect on quite a lot of the core genes, and even though those are tiny, they can actually add up to be non-trivial perturbations. But it is important to note that sometimes in this upper model, you can have a trans-regulator that does regulate a lot of core genes in a really coordinated way, and these can actually have important effects. And there are several really quite clear examples of this in the literature, and I'll just close with one of these. So this is a really beautiful paper from Mark McCarthy's lab, led by Karen Small, published in Nature Genetics a few weeks ago. And this is a paper about a gene called KLF14. And so KLF14 has a cis EQTL, and this cis EQTL has been found to be associated with lipid levels, with obesity, and with diabetes. And so in trying to understand what's going on here, they learned that this cis-EQTL is also a trans-EQTL for a network of 385 other genes that are specifically in adipose tissue. And this picture is just an illustration of these 385 genes that it's a trans-EQTL for. And so basically what it does is it has important effects on the differentiation of adipose cells. And what they actually believe is that several of these genes that are in this co-regulated network seem to be really good candidate genes for actually driving the different phenotypes that the KLF14 variant is associated with. And so I think that in this setting what you have is a variant that's a cis-regulatory variant to the KLF14 gene that is a trans variant to a whole series of what I would describe as core genes that have direct causal arrows to the phenotypes that they're interested in. So when we think about the SNPs that wind up at the top of the list in a GWAS study, this is going to be a mix of different kinds of variants. Some of those are going to be in cis to core genes. Some of those are going to be variants that are close to peripheral master regulator genes, and occasionally, occasionally there'll be bad luck where you've got a gene that doesn't really do a whole heck of a lot involved in the disease, but the signs kind of add up in a malicious way to produce a signal almost by chance. Okay, but most of the time the signals that are near the top of the list will actually be useful to dissect molecularly. Okay, so I'll end there with a few conclusions. So first of all, we'd like to propose that gene regulatory networks are really highly interconnected, such that all the genes that are expressed in disease-relevant cells are liable to affect functions of core disease-related genes, and that most of the heritability is due to SNPs that sit outside of core pathways and act through transregulation. And we refer to this hypothesis as an omnigenic model. Okay, secondly, I've tried to argue in the second half of this talk that the model is consistent with what we know about cis and trans EQTLs. I showed you that trans variation is responsible for about 70% of expression heritability, but the effect sizes are nearly uniformly tiny, which means that even though we can't find them yet, there must be an enormous number of trans-acting EQTLs. And I like to think of co-regulated gene networks potentially as acting as amplifiers for this peripheral variation. And then the last comments that I want to make are about the future of GWAS in light of our model. So first of all, I think it's important to consider what are the purposes of doing gene mapping. And I would argue that there are two main goals. So the first of these is genetic prediction. So if you see somebody's genotype, can you predict their risk of disease? And for a long time, that's been a really difficult goal, but there has recently started to be a lot of progress on this as sample sizes have become bigger. So there's a paper from Seth Cathareeson's lab recently that I really like that shows that in the case of coronary artery disease, that the top 2 or 3% of the polygenic risk score distribution are actually good candidates for being put onto statin medications, and so they would argue that in that case, polygenic prediction for coronary artery disease has actually reached the point of having translational potential. Now, for genetic prediction, doing GWAS is really absolutely essential, because what we need to do is to get good estimates of those very, very small effect sizes. And we don't actually really care whether a SNP is core or peripheral, what the mechanism is. We just need to measure effect size as well. And luckily, there are a lot of efforts now with integrating genetic data with electronic health records and big biobanking studies. So I think that the next phase of very large genome-wide association studies are going to be much, much cheaper and easier in the context of other activities than the last 10 years. Okay, but the second goal of doing gene mapping is more challenging, and this is this is the goal of identifying core genes and core pathways and master regulators that might be good druggable targets. And I think that our model is suggesting that just because something's a good hit in the GWAS study doesn't automatically mean that it's going to be a useful thing to study molecularly. And my thoughts on on how to proceed with this are a bit complicated, but I think that the next stages of the field are going to involve some combination of really deep exome sequencing to find rare variants with larger effects. More GWAS is really going to be helpful for this in terms of getting better estimates of effect sizes. And lastly, I've been very interested in getting better methods for network inference and also methods for layering new estimates of gene regulatory networks on top of the GWAS data, because ultimately what we'd like to be able to do is to partition genes into the set of core genes such that if you know the regulation of core genes, then the disease risk is conditionally independent of everything else. So I'll close there, but let me just thank the people who are involved. So Evan and Yang and Chuan Yao have done an enormous amount of work, and both in terms of data analysis and talking through all of these models. So I'll acknowledge their contributions as well as my labs. So here's a picture from ASHG a couple of years ago where we had some alumni, and so my whole lab's been very useful in talking about all of these issues. So I'll close there. Thank you for your attention.